All right. Thanks a lot, Alex, for that introduction. Thanks for having me here. Indeed, the thing I'd like to discuss today is how we can learn the dynamical laws of migrating cells from observations. And I want to do that from the level of individual cells all the way to multicellular collectives. And just to warm you up, I want to just start with a couple of examples that really inspired us to think about many of these problems. So what I'm showing you here is some examples of immune cells. And for cells like these to be able to perform their function, they need to navigate these complex extracellular environments. And to get around effectively, cells like these have to constantly solve migration challenges. So for instance, the cells here at the bottom, they need to make it towards this site of inflammation. But to get there, cells need to traverse these long constricting channels of an intravascular network. This cell above here, to get around, it needs to squeeze itself through pores that can be sometimes substantially smaller than the nucleus of the cell. So cells like these can really navigate through these environments in amazing ways. And the thing that we would like to understand is how cells really dynamically resolve such migration challenges. Another interesting challenge arises when cells, and you see this happening a couple of times over here, when cells start colliding with each other. So when cells engage in making contact, one cell can really qualitatively affect the dynamics of how the other cell moves. And this is going to become particularly relevant and interesting in a denser, larger multicellular systems where frequent interactions between cells could give rise to all kinds of collective behaviors. And I'd argue that if we really want to understand how cells can control functional multicellular migration, then we need to figure out the dynamic rules of engagement between these cells. Now, a lot is known about the underlying molecular machinery, but there's also a problem because even if we know what each and every one of these molecules is doing, it is a tremendous challenge to integrate this information and derive an effective dynamical description for how a cell at the systems level is moving, how it moves as a whole, how it interacts with its environment and moves as a response of all kinds of inputs. So over the last couple of years, we've been exploring a completely different approach to this problem, which starts with a very simple question. Could we learn a dynamical description of moving cells from data? And we'd like to do this in such a way that we can actually arrive at a physically interpretable dynamical framework. So a dynamical framework that could in principle couple down to the, or link down to the underlying molecular machinery, and a dynamical network that is interpretable in the sense that we could disentangle the innate behavior of a cell from how a moving cell would couple to its environment or how it responds to interactions with other cells. So in going about this over the last couple of years, we tried to do this as systematically as possible. So we really started just by looking at individual cells, then pairs of interacting cells, and now larger multicellular systems. And I'm kind of going to take you through this logic as we went through it. And all of this starts with an experiment that really inspired us a lot. And all these experiments come from the lab of Joachim Redler at the LMU in Munich. So Joachim and his lab are really experts at creating artificial microenvironments for cells. And one of the environments they created is this kind of cell in a box experiment. It's a dumbbell-shaped micro pattern. So the idea of these micro patterns, they're just two-dimensional patterns, but they're coded in an interesting way. So everything inside the white boundaries is coded with fibronectin. So cells can form focal adhesions, they can adhere to the substrate, but everywhere outside the white regions, this is like the forbidden region for these cells, the substrate is coated with PEG, and the cells cannot adhere. All right? So you can see that the cells are effectively confined, but they're very dynamic. They constantly form protrusions, they change their shape, and you can see that the cells can quite effectively migrate across this constriction and solve this migration challenge that we designed for it here. Now, there's many ways you could imagine, and that we also explored in analyzing data like this, but the thing we started out with is really the simplest thing you could imagine, which is just to track the center of the nucleus over time. This gives you these position time trajectories. Okay? Obviously, when you do this, you lose a lot of information, but it captures in a very low dimensional fashion how the cell as a whole moves through this confined environment. Now, the beautiful thing about these experiments is that they're high throughput in nature. So we quite easily gain access to a large ensemble of such trajectory data. Like over the years that we've been working with Joachim in his lab, he's really been generating literally hundreds and thousands of hours of cell migration data with different cells, different drug treatments, different genetic perturbations, just generating an enormous amount of data that gets me as a statistical physicist really, really excited. Now, as I'm talking, you're probably staring a little bit at these trajectories, and you might notice that there might be something stereotypical about how these cells move through this confinement, but there's also a great deal of variability. So one of the first questions that we wanted to solve is whether we could actually disentangle deterministic from stochastic contributions to cell migration dynamics. And in essence, what we're seeking then is something like an equation of motion for confined cell migration. Now, in analogy with something like a Brownian particle in a liquid, what we're really looking for then is something like a stochastic equation of motion or a Langevin equation. For the Brownian particle, there would be something like a deterministic force law, Stokes' drag law, and then there's going to be a dynamical noise that is related to the drag, but is also controlled by temperature. Now, for the cells, we do not know the deterministic law. We do not know what controls the noise. And to be really honest, we don't even know for sure what the right structure of the equation of motion is or the right order. And you can make different arguments here, right? So you could argue that, the cells are all operating at low Reynolds numbers. So what we really should be considering is something like an overdamped equation of motion. And you can make arguments that you could somehow expect that there's going to be contributions that relate to friction processes or dissipative processes, something that depends on velocity, and maybe another term that derives from a force term that derives from a potential. But you could also argue that a migrating cell is really an active system. And in an active system, activity can introduce a new time scale that essentially creates an effective inertia in the problem, even if you're at low Reynolds numbers. So maybe we should really be considering underdamped equations of motions. You can make another argument that the cytoskeleton in the cell and many other aspects of the cell have very interesting mechanical properties. It's a viscoelastic system. So really there should maybe be lots of different timescales that are relevant for how the cell moves. And so maybe we should also be considering memory kernels and things like that. So really, you can make very valid arguments for very different kinds of equations of motions that one should be considering here. So this forces us to be really agnostic in our approach and to use a data-driven theoretical approach to essentially select the appropriate model based on the data. So let me walk you through how we did this. So for instance, if you consider this equation of motion in black here, this is the underdamped Langevin equation. What it would state is that the acceleration of a cell is driven by a deterministic driving term, this F, which depends on the position and the velocity of the cell on this micropattern. And then there's a dynamical noise that we could assume to be Gaussian white noise, right? This could be one possible model. So how could you investigate whether this is reasonable? Well, what we do is we take the trajectories, and essentially we're going to use short timescale information from those trajectories, positions, velocities, accelerations, to somehow infer the structure of this Langevin equation. And what we do in practice is we take, for instance, the force field in the Langevin equation, and we decompose it into a set of smooth basis functions. And here you can use whatever makes sense for your problem. You can make use of the symmetries to select, for instance, Fourier modes or polynomials or anything else that somehow makes sense for your problem. And then what we did is we derived unbiased and robust estimators for the projection coefficients of those basis functions, basically the C alphas, using the short timescale information. And out comes something like a deterministic driving term. Okay. Now, this doesn't mean that this is the correct model, because we could do this for many different classes of models. You can take the data and you run the inference and something will come out. Doesn't mean that it's right. So you have to do validation. So how do you do validation of these models? You take the model and you simulate it. And so you get, simulated trajectories of hopping cells. And then what you need to do is you need to make predictions with the model for statistics that you didn't use to constrain the model in the first place. So when we do this for many different classes of model, like the overdamped equation of motion, we then arrive at predictions that really fail to capture experimental measurements of those statistics. But when we do this for this underdamped class of models, we find that a model constrained on short timescale information can capture quite adequately long time behaviors like steady state probability distributions of position, velocity, a survival probability, the probability that the cell has not yet made a jump after a certain amount of time, a velocity correlation function. And we can also show that these inferred models can accurately capture the time scale of this process, the time it takes for the cell to make a transition, even if we vary the length of the bridge by roughly an order of magnitude. So what this tells us is that this underdamped Langevin equation is the appropriate structure to capture cell motion on confinement from roughly tens of minutes, which is the resolution of the experiment, to up to about 48 hours, the time it takes for the cell to divide. Now, the model we infer also lends itself to interpretation. So when we first, were staring at data like this, we were really wondering how we should be thinking about a problem like this. And intuitively, what we thought is that the constriction acts a little bit like an energy barrier, because the cell needs to deform to squeeze itself through this constriction. And then we wondered about the role of the noise and maybe, facilitating transitions across such a barrier. And these pictures are not very different from, pretty common models used to describe cellular dynamics in multicellular systems. But the model we inferred from the data for this cell here actually points to a very different conceptual picture. So here I'm showing you the nonlinear dynamics of the deterministic term, and it exhibits limit cycle oscillations. And these limit cycle oscillations already describe how the cell makes the transition. So this is really encoded by the deterministic dynamics of the cell. The role of the noise is not to facilitate a transition, but it actually plays a role in speeding up the transition quite significantly. Another interesting feature that you can kind of see from these phase portraits is that the cells deterministically accelerate when they move through these constrictions. And this turns out to be a very robust feature of many different cells and many different confinements that we first saw when essentially inspecting such an inferred model. You can also see what happens when you remove the constriction. So if you remove the constriction and then again infer the model, you see that a lot of these features that we associate to this constriction indeed vanish. Now the nonlinear deterministic dynamics just as a line of stable fixed points at zero velocity, and you don't see this deterministic acceleration. You don't see limit cycles anymore. Right. So with an approach like this, you can now start exploring all kinds of generalizations of this problem. You can look at cells in different geometries. You can look at different cell types. And we've been exploring this quite extensively. But one generalization that I really want to focus on today is what happens when you just wait long enough for the cell to divide such that you have two of them on this pattern. And then you arrive at an experiment that we'd like to think of as our cell collider experiment. Because just with this dumbbell-shaped geometry, you generate these discrete collision events between cells. And if you look at this example here, you can see that these collisions are also very variable in nature. So sometimes the cells, when they form protrusions and these protrusions hit each other, the protrusions tends to retract and the cell reverses. Sometimes you see that the cells kind of slide past each other, exchanging positions on the pattern. And occasionally you also see that the cells kind of join together and start following each other across this pattern. Now, to analyze this kind of collision behavior, we decided to also take a broader perspective, looking at many different cells in parallel. So I'm showing you a whole range of 5 different cell types that we've been exploring, running sort of from a mesenchymal phenotype. So this is a human breast cancer cell type, a cell that does not express EC adherens. When you put it on a two-dimensional substrate, it doesn't form confluent layers. And then as we move towards the other end of the cell, so some are healthy, some are cancerous cells. All the way on the other end, we have human epithelial breast tissue cells. These are cells that express EC adherens, and when you plate them on a two-dimensional substrate, they form these confluent layers. So what we can then do is we can take pairs of cells, they're always the same cell type, on the cell collider, let them collide with each other repeatedly, and measure lots and lots of statistics of how these cells interact with each other on these patterns. And already just looking at these trajectory data, you might see that there's some very interesting differences between the distinct cell types. One difference that I want to highlight is in the type of collision behavior that dominates. So if we look at the human breast cancer cell, so the mesenchymal cell, this one predominantly slides past the other cell when they collide. If we move towards the other end, the epithelial cell type, these are cells that when they run into each other, they predominantly tend to reverse. Okay? So there's just one clear distinction between these different cell types. So what we'd like to do next is figure out how to conceptually think about the underlying dynamics that is driving these kind of distinct behaviors. So again, we'd like to infer something like an underlying equation of motion. Now it's going to be an interacting stochastic equation of motion of two pairs of cells. We went through a model selection procedure, and this allowed us to arrive at a model that is a fairly intuitive and straightforward generalization of what we just had for an individual cell. So what do we see? So we have a one-body term, as we had before, just explaining how one of these cells interacts with this confining pattern. We have a dynamical noise, as we had before, but now there's also two interaction terms that we want to consider. So one of them couples to the separation between the cells. We like to think a little bit like this as a cohesive-like interaction. There's a function in front, f, little f, which can also depend on the separation between these cells. And this really characterizes the cohesive interaction. It characterizes its range and its sign, whether it's more attractive or cohesive. And then there's an interaction that couples to the relative velocity. And this is a little bit like a friction-like interaction. Okay, and again, there's a prefactor in front, this little gamma, which depends on cell separation, which can characterize the range of this interaction. All right, so we take the trajectory data, we perform our stochastic force inference for this model using trajectory data of interacting cells, and then we just extract the inferred terms. So there's two big surprises that I want to start with. The first surprise is actually the one-body term. It is pretty much identical for what we infer for these cells when they're just moving by themselves on the pattern. And this is not at all obvious to me because it could have been possible that the presence of the other cell could have led to some kind of adaptation for how the cell is interacting with its confining environment. But we really don't see that. The presence of the other cell really only manifests in the interacting terms. So when we infer the interacting terms, what we're really inferring is the structure of these functions, f and gamma, which characterize these interactions. So for this particular mesenchymal human breast cancer cell, we see that there's long-range attraction with long-range repulsion with short-range attraction. But the big surprise is really in the friction-like term because the sign is off, right? It should be negative, but we see a positive sign, which means there's like an anti-friction behavior, a contribution that accelerates these cells past each other in a way that actually increases with their relative velocity. And this really reflects the fact that this is an active system and the underlying machinery that controls these interactions is active. So we did this for all 5 cells in parallel. And I'm first just going to show you what we observed for the one-body term, like how just an individual cell responds to its confinement. And here, actually, the conclusion is very simple. It's pretty much the same for all cells. So this way of how the cell moves through its constriction turns out to be very universal across this very broad range of distinct cells. Cells just really want to accelerate through the constriction. The big differences are mostly reflected by the two-body terms. So let me just walk you through this. On the mesenchymal end, so that's the one I showed you on the previous slide, we see short-range attraction combined with anti-friction. And then as we move towards the other end of this range, the epithelial end, we see that this slowly crosses over to more repulsive kind of behaviors, combined with a more regular friction interaction. Now, the models that we infer here are high dimensional, and this makes it sometimes a little challenging to compare them side by side. Also, if we're interested in looking at all kinds of molecular perturbations or drug treatments and all that. And therefore, we decided just to make a simpler comparison between different behaviors is to perform a dimensional reduction. And we can do this here in a very simple way by just taking the dominant contribution to the cohesive and the friction-like interactions, let it span a two-dimensional plane. And this plane we like to think of as our interaction behavior space, because depending on where you are in this space, you can pretty much predict what the dominant collision behavior is going to be. So if you're in the upper half plane, the dominant collision behavior is going to be sliding behavior. If you're in the lower right quadrant, the dominant behavior is going to be reversals. And here are the 5 different cell types scattered over this plane. So, yeah.
Right. So in your reduction, you're basically taking those functions that you have of delta x and turning them somehow into a single number.
Yes.
Can you comment briefly on how you do that?
Yeah. So, when we consider those functions, we do basis expansions. Yeah. And basically what we're taking is something like a leading order term in that expansion and just saying, let's just only take that number. Of course, that's so it's a very crude approximation of the full model, but it turns out to be quite predictive of the dominant behavior. All right, so we see big differences between, for instance, a mesenchymal and an epithelial cell type, which I don't think would surprise anyone. But this is a way to quantify those differences and to figure out really what those differences mean. And I think that is where data-driven theory can be helpful, to just analyze heaps of data in an unbiased way and figure out a way to quantitatively and conceptually extract something useful from it. But what it does not immediately give you is something like the underlying mechanism. And we'd like to push a little bit further to see if we can understand where these kind of different behaviors come from. All right, so to figure out what might be a reasonable underlying mechanism, we took a step back, looking at some of the original data just of one cell on these patterns. And, I think this movie already shows you that if you only take something like the nucleus coordinate, that you're missing a lot of information. Obviously, these cells tend to be internally polarized. There's a back, there's a front. This determines where the cell forms protrusions. These protrusions play a massive role in guiding the cell through this constriction. So we were then wondering, could we maybe not slightly expand our dynamical description to start incorporating information like this? For instance, imagine that we could write down something like a dynamical model for a slightly expanded set of dynamical degrees of freedom, not only the nucleus now, but also something like a protrusion coordinate and the internal state of polarity. Okay? We could write down a tentative model that would couple these degrees of freedom. We don't know whether this model makes any sense at this point, but we can write it down. And these functions, little f, g, and h, that's where all the biophysics is. This is where, this is what couples all these degrees of freedom. Also note here that the role of the polarity dynamics in this model is to drive the protrusion coordinate. Okay, this really generates the active force that drives the protrusion coordinate. So we've been working on trying to figure out whether this model makes sense, just from a bottom-up approach, trying to derive what these functions f, g, and h look like, and we made progress. But actually, the first thing we did was a data-driven approach. Just say, let's take the data as it is and try to see if we can constrain what this model should look like. So for that, we needed to automatically segment the cell shapes. And from the cell shapes, we extracted something like a protrusion coordinate, which is shown here in green, something like the center of mass of the protrusion. Here you can see an example of protrusion and nucleus coordinates as the cell is moving over this pattern. And we did this for a very large number of trajectories. Using those trajectory data sets, we constrained or inferred the underlying model. Yeah, Daniel.
Given this, should I understand the previous model to be like the data was not conditioned on whether the cells were contacting? It's just like literally whatever their center of mass separations were, regardless of whether they were physically interacting?
Yes, yes. So this is also why the interactions tend to have just a certain range. That range corresponded to a typical cell size, but we did not condition the model on making contacts. That's absolutely right. Because we did not have information about a protrusion coordinate in the model. I'm going to show you in a second how we could use this also to study the interactions a bit more. All right, so I'm not going to show you the details of how we did this inference, but I'm just going to fast forward to the final result and try and convince you that there's something interesting to be learned by this. So this is what the inferred model looks like. It is intuitive in the sense that you see that there's something like an elastic coupling between the nucleus and the protrusion coordinates. There's something like a confining potential that just makes sure that the cell doesn't run off the pattern. But the really big surprise, and I think the most interesting result from this inference, is in the dynamics of the polarity coordinate. So why is it interesting? So what we see in the inferred model is that the polarity dynamics is very sensitive to where the protrusion is on the confinement, especially when the cell is on one of the square islands. What we see is that this alpha here, because it depends on where the protrusion is a positive number. This means that the polarity has a negative feedback onto itself, and the cells do not want to spontaneously polarize on the square islands. This changes when the protrusion starts moving into the constriction. So the inferred model tells us that somehow the cell knows about the confining geometry that the protrusion is experiencing, and as a result of this, the feedback of the polarity onto itself goes from negative to positive feedback. So basically the sign of this alpha changes. As a result of this, there's a bifurcation in the polarity dynamics. The cells now want to spontaneously polarize. Yeah.
Does one know what is first? Is it the polarity driving the cell, or the cell is squeezing in, so now the volumes are very low, and there are a few new enzymatic reactions happening that is drawing the polarity system there?
Yeah, so if I understand your question right, I mean, this model does not make a statement about what is generating this feedback mechanism. It just says, well, if we want to make sense out of all these different statistics of the protrusion and the nuclear coordinate, a model that can capture all these statistics is one in which you have this feedback mechanism. It does not tell you what the underlying molecular process is or you know what has caused this. That I think is still an open question.
Dynamic movies. So there would be scenarios where it failed, like the process begins and then it fails.
Absolutely,
So
yeah.
just look at segregated movies of that kind, where the process reversal happened, and learn what comes first. I don't know if this
Yeah, so actually that's a good point. You have a lot of inherent stochasticity, right? Sometimes the cell tries to form a protrusion also on the bridge, but then it retracts again. Actually, those kind of failed attempts are also captured by this model. So there is inherent stochasticity, and we have to be able to capture also that variability. So that is a constraint that we're using to constrain the inference. So in that sense, you're right. We take that into account. All right. So why am I emphasizing this so strongly? Because we were very excited when we saw this, because we thought this is, I think, a really nice example of how you can use data-driven theoretical approaches to discover a potential biophysical mechanism of what a cell is doing. That it's using some kind of adaptation mechanism, a geometry adaptation mechanism for this protrusion and polarity dynamics that facilitates a cell in moving through a constriction, because it's this nonlinear dynamics, this bifurcation here that leads to this very characteristic acceleration of the cell through this constriction. And then you can do all kinds of things now to figure out what the underlying molecular mechanism is. I'm not going to dive into that now, but now you can do drug treatments. You can treat the cells with latrunculin, with blebistatin, ROCK inhibitors, and try to figure out what about this polarity mechanism is giving rise to this behavior. And then you can do model inference in all cases and really try to get insight into the underlying mechanism. What I want to do instead today is get into Daniel's question about interactions now, because you can also take this model where you now have information about a polarity and a protrusion coordinate, and you can generalize the model. So far we believe that this is an adequate model for a single cell. What about interactions? We do not know how to derive these interactions from the underlying molecular machinery, but what we can do is guess. So we use the phenomenological approach here, where we just said, well, we have all these degrees of freedom. Let's just couple them together in all kinds of different ways. We can use symmetries to rule out many, many possibilities. And we can, only consider leading order contributions to keep the model simple. If we do that, how many possible candidate interactions would we be left with? Actually, quite a number, 14 in total, if we count it correctly. And so these are all possible interaction mechanisms. So let me just give you one example to give you a little bit of intuition for what this could mean. So there could be one interaction mechanism in the blue equation of motion for the nucleus, which couples to the two nuclear coordinates, basically the two cell nuclei pushing on each other. Maybe there's an interaction mechanism that has that form. But there could also be many others. Now, in principle, if you have a model where you write down terms that are allowed by symmetry, if they're allowed to be there, typically they are there, right? So this could be a very daunting situation where you might need a model with 14 different interaction terms, and that's probably not going to be very useful. But maybe it is quite different. So we didn't know, and we just wanted to use the data to figure out which interactions are needed to describe these interacting cells on the micropattern. So we went for a brute force approach, a statistical inference that just considers many different combinations of all these possible interaction mechanisms, and then just try to figure out which combinations are needed to capture the data. So we looked at many different statistics to do this inference. I'm not going to show you all that. I'm just going to show you the summary in this interaction behavior space. So that's the model that in the end we arrive at. It only has two interaction mechanisms. So only two are needed from the 14 possibilities. Both of the interaction mechanisms drive the polarity dynamics of the cells. That's another interesting finding. The first one is an interaction mechanism that arises when the two protrusions bump into each other. And what happens then is the cells just tend to depolarize themselves. The interaction parameters that are associated to this are pretty much the same for all the 5 cells. So this is not the mechanism that distinguishes cell behavior. It's this one. It's a polarity-polarity coupling mechanism, an interaction driven by the polarity differences between the two cells. And the sign and magnitude of that interaction mechanism is really what distinguishes the mesenchymal from the epithelial behavior. And what you can then do, and this is the next step, you can start considering all kinds of molecular perturbations. We kind of want to see if we could break the model by taking an epithelial cell and inducing EMT using TGF-beta or blocking efferents or E-cadherins. We also started from mesenchymal cells and started expressing E-cadherins. And basically what we see is that we can get a mesenchymal cell to look more epithelial and the other way around, but the behavior always just moves along the behavior predicted by this model that we inferred from the data. All right, so in the last couple of minutes, I want to talk about multicellular behavior, because if we want to figure out cell-cell interactions in relevant context, we need to be able to do this for large multicellular systems. It's still a little bit a simplified system, so we have one-dimensional strips, but now we're going to let the cells proliferate. So these are the epithelial cells. You can see that as we get more and more cells, they tend to cluster together, and the clusters are very mobile. So they tend to move coherently, a little bit like a flock. And that's the kind of behavior we typically see for these human breast tissue epithelial cells. Just for contrast, I want to show you a fibrosarcoma cell, a mesenchymal cell type. You see here something very different. First of all, you note that the trajectories hardly ever intersect each other. They also tend to avoid each other a little bit. Sometimes you do see that the cells form from clusters, but these clusters are not very mobile. They're quite immobile. And then, as a third example, the human breast cancer cells, again a mesenchymal phenotype, very different behavior. So these cells occasionally do tend to do something that looks like flocking, but it's not as persistent as the epithelial cells do. We also see a lot of exchanges, cells sliding past each other for this cell type. So just for these three cell types, we see very different behaviors on this very simple geometry. And again, we wanted to figure out what this means. So one of the things we first looked at is like general statistics to characterize their collective behavior. Think about velocity correlation functions, and you see big qualitative differences. So the epithelial cells have mostly positive correlations. The two mesenchymal cells have negative correlations on short scales, positive correlations on longer scales. We look at pair correlation functions, something like the fraction of time that the cells spent in a flock or just in a cluster that is not flocking. And we see differences. And these differences also depend on the packing density of the cell. So this is just a way of characterizing collective behaviors. But can we figure out the underlying dynamical rules? So we did the one body problem, the two body problem. So now we want to do the many body problem. We want to do many body stochastic inference where we have pairwise interactions between the cells, but we don't know what the structure of these pairwise interactions is. We assume that they depend on dynamical variables like cell position and velocity, but we want to be as agnostic as possible about how to figure out these interaction terms. So what we did is we set up a pipeline for a procedure that does this in a more or less principled fashion. So what we're considering here is many possible classes of interaction model, where each class is defined by a particular dependence on dynamical variables and certain symmetry properties. And they're just listed here a little bit in order of increasing complexity. For each possible interaction class, we do a model inference. So we assume the interaction class, then we take the trajectory data from the multicellular system. We use this underdamped Langevin inference algorithm to infer the force fields. Then we take the model, and now we arrive at one model for each interaction class, and we then make predictions for the model for statistics that are somehow independent of the inference, like collective statistics like the velocity correlation function, but also clustering statistics and stuff like that. And then we say, well, the model makes a prediction. We can compare that prediction to an experimental measurement for those statistics, and we can characterize it in a single number, the R squared. And if the R squared is 1, it means the model is really right on top of the data. If it's 0, we kind of get the averages right. If it's less than 0, it's even worse. So just running this pipeline for these three different cell types, we find the following result. So first of all, you notice that many models are really bad. But some models come out as like clear winners. They're not perfect, but they do really well. Let me just show you what the winning models show for these three different cell types. So many aspects reflect what we already learned for these cells by just looking at pairs of cells on the pattern, which is somehow satisfying. But there are some additional insights. So one thing we for instance, see is that, okay, for the epithelial cells, we saw as before that there's kind of a repulsive interaction and something that looks like friction, which you can also think of as an interaction that tends to align the velocities of the two cells. For the two mesenchymal cells, you see something a little bit more interesting. There is, on short length scales, the cells tend to anti-align their velocities, and at longer length scales, they tend to align their velocities. But the big surprise is really that we needed one additional contribution to these interactions that we hadn't detected for just pairs of cells. And that interaction is one which you can think of as an additional term to the dynamics, which depends on the velocity of the cell itself. So if a cell bumps into another cell, depending on proximity, there's going to be an interaction that is mainly driven by the velocity of the cell itself and not by what the other cell is doing. And this interaction is absent for the epithelial cells, but for the two mesenchymal cells, we see it, but it has an opposite sign. So for the fibrosarcoma cells, what this interaction is basically doing is when a cell bumps into another cell, it's like it takes its foot off the gas, keeping the steering wheel straight. It just decelerates along the direction in which it's moving. The human breast cancer cell does the opposite. When it bumps into another cell, it hits the gas, it accelerates into the direction in which it was already going. Now, why is this interaction interesting? I think for two reasons. The first one is it's a non-reciprocal interaction, as opposed to all the other ones that we considered so far. And this non-reciprocal interaction could allow cells to access collective behaviors that are richer that you could achieve with reciprocal interactions. And, for instance, what this non-reciprocal interaction is doing in this system is it's allowing the cells to control their flocking dynamics. Essentially, what this interaction can do is it can tune the speed of the flock in a way that is independent of the individual speed of the cells. So with the velocity inhibition, what we saw for the fibrosarcoma cells, you can get the flock to move much slower than the speed of an individual cell. With the interaction we saw for the case of the mesenchymal cells, in principle, you can get the flock to even move faster than the speed of an individual cell. So this non-reciprocal interaction allows you to defy the common wisdom that, we always say that if you want to go far, you have to go together. If you want to go fast, you should go alone. This is not the case for these mesenchymal cells. They can actually go faster by going together. And that's an interesting aspect of this interaction. Let me finalize here. I want to just mention all the experiments that I showed you were done by Joachim, Alexander, and Emily. The theory work was started by David and is now continued by Tom and Bram. Bram is also here in the audience, and he did all the work on the multicellular inference. Thank you very much.
I just had a question. So you showed all this stuff on the confined environments, and you said that it really depends on what kind of environment you have. What do you think the promise of this is for imaging the interaction potentials in a developmental system, like you showed at the beginning, if it's so sensitive on the nature of the environment?
So what we did is we looked at different environments, and what we saw is that even though like the one body term changes if you go to different environments, but the interactions actually remain mostly unaffected by small changes in the environment. What we also did is we took the interactions that we learned from like simple assays like this, and then we make predictions for what you see for like two-dimensional confluent layers. And actually the predictions are pretty good. So there is promise, but if you want to take this to developmental systems, there's a few additional challenges we need to worry about. I mean, there's dense packings. We might have to worry about cell shape, which we're not really including here yet. There could be different cells interacting with each other, which have different mutual interactions. So all of that we haven't considered so far. But as an inference problem, I think there is promise to go there.
1 second.
Really nice. I was wondering, I guess related to that, you said your devices were fibronectin, but what about if you kind of change the matrix, make it less appealing on the sides than for kind of other, matrix that might have different adhesion profiles?
Yeah, that's a great suggestion. We didn't explore that so much. We explored different geometries and different, concentrations of fibronectin, but we haven't explored different kinds of chemical patterns. That's a great suggestion.
Really great talk. Thank you. I was really interested going from the single cell to the multi-cell to like the multi-body problem. Do you anticipate that going from multi-body but kind of sparse to a confluent tissue, the same sort of approach will still hold of kind of having this interacting particle model? Or do you think that there's going to be some limit where it breaks down in the continuum?
Yeah, so I think you can get a long way, but I think if we really want to understand that, we would like to go to a representation that can capture vertices and things like that. So I think that is where we ultimately have to go. But I think what we've noticed so far is we can push this particle representation pretty far. And we haven't seen it break yet, but I do expect it will break if you're looking at two-dimensional confluent packings and stuff.
I was just going to piggyback on Margaret and ask, in the context of different cells, have you started mixing some of your cell types, or one's labeled so that you know which one's which or something?
We haven't yet. I mean, that's a very exciting direction, but it's just experimentally a little bit tricky. So we haven't explored it. Yeah, I agree.
One question from there.
Oh, we don't have-
So two quick questions. The first is you saw this acceleration that's happening through the subscriptions. And I wonder if the acceleration is just intracellular acceleration, not the cell body itself accelerating, just because the nucleus motion could be different from the cell body moving forward, right? Because of the differences in cell motion. So perhaps the cell body is constantly moving at the same velocity. The nucleus could be accelerating because of the nuclear piston.
So we did a few different things, like also taking just not the nucleus position, but something like the center of mass of the cell or other coordinates. And all of these features are quite robust to what you decide to use as your, center of the cell. So it's not entirely depending on using that particular choice.
Well, then so you saw these two dominant mechanisms of polarities being reversal versus polarity. Is that a different way of phrasing the CI, the contact inhibition of locomotion versus Wittek-like alignment versus?
Yeah, so that's a very interesting point, right? I mean, we've been using all these qualitative descriptors of cell behavior. I mean, contact inhibition of locomotion is like almost 70 years old. And it's actually not obvious for me how to map what we're finding here as quantitative dynamical rules on that. To some extent, yes. But for instance, I'm not sure whether it's really about these polarity-polarity interactions that we talked about, or maybe this non-reciprocal term that I was discussing towards the end, because many of these have somewhat similar qualitative effects, but if you look at the quantitative model, there's still big differences. So I think there is a mapping, but it's not straightforward.
Can go.
Yeah, great talk. I was wondering, can you use for the collective case for hydrodynamic style continuum scale models, which are
Yeah. So yes, in principle, you can do that, but we haven't explored it. So you could, in principle, take these interactions, perform some kind of coarse graining procedure to derive the effective hydrodynamic descriptions.